Okay, well, welcome everybody. Thank you for coming back to another installment of the Alaska Wildlife Alliance's Wildlife Wednesday. I'm really excited about our talk tonight, learning about hormones in whales. But first, I've got some virtual engagement guidelines. Next slide, please, Valentina. Because we are using Zoom, we want to be very careful that we don't have any unintentional Zoom bombing. So we're going to make sure everybody keeps their video and their microphones in the off mode during the presentation. This will also help us with our connectivity strength and to save some bandwidth. If you're new to Zoom, we encourage you to view the presentation in full screen mode. So look for that little picture frame symbol, and that should help you toggle between different views. And if you want to ask a question, Valentina is going to give her entire presentation, but you can feel free to ask your question at any time using the chat feature. So you can go ahead and type it. We'll wait until she gets through her presentation, and then we'll go through the questions at the end of the talk. But feel free, if a question comes to you, go ahead and put it in the chat at any point in time during the presentation. And the most important thing tonight is we hope you enjoy this talk. We hope you learn something new, and it's a fun, family-friendly event. Next slide. Before I get into Valentina's presentation, I do just want to introduce you to those of you who are not aware of who we are. My name is Mandy. I am the Deputy Director of the Alaska Wildlife Alliance, and AWA is an Alaska-based nonprofit. We were founded right here in Anchorage in 1978, and we work to protect Alaska's wildlife through citizen mobilization, advocacy, and education, such as our Wildlife Wednesdays series. We would love for folks to join our alliance by getting a membership for as little as $35 a year. You can visit us on our website at akwildlife.org. Or there's other ways if you like what we do and you want to support our work. We are a charity on several different organizations. We are set up to receive donations through Facebook, through Amazon Smile. If you do any shopping on Amazon, we encourage you to go to smile.amazon.com instead and select your charity. It's the same price for you, but Amazon will donate a percentage to our organization. We're also working with Roundup app. So if you make a purchase through the app, they'll round up your change and donate it to us. We're a charity on Pick, Click, Give. And for those of you who might be or know federal or military personnel, we are part of the Combined Federal Campaign, the CFC program, and that's going on right now. So we would love your support in any way we can. Thank you. And next slide, Valentina. We just want to let you guys know that we do use social media. If you're not following us, here's just a quick hint of some of the things that you're missing. We make posts about wildlife advocacy opportunities and updates, and I'll give you a couple of those here in just a minute. We post, our Wildlife Wednesday series and other educational activities and events on there. And then we also like to have a little bit of fun. If you're not on social media, you can go to our website to sign up to receive our monthly email newsletter, and you don't have to be a member to receive those. Next slide. So one of the big advocacy things going on right now that you still have a chance to get your voice in and be a voice for wildlife is to put in a comment regarding the U.S. Fish and Wildlife Service's proposed regulations, which would roll back wildlife protections on the Kenai National Wildlife Refuge if those regulations are approved. So they're accepting public comment until November 9th. But some of the things, if they're approved, these rollbacks will allow killing brown bears over bait, and they won't have a requirement for the meat to be salvaged. They allow for more inhumane methods of fur bearer trapping, and then they will also allow for traps and snares near hiking trails and campgrounds. So there's some very concerning things in this proposed rule. And you can learn more about this topic by going to our website, akwildlife.org, and we have a search bar at the bottom of the page, and you can just type in Kenai rule, and that will give you more information, a fact sheet, and also tell you how to go ahead and comment. If you're interested, we also have a text service where we send out alerts, and if you text the word refuge to 833-541-0408, it'll sign you up to receive those alerts. And I'll go ahead during the presentation and I'll put that phone number in the chat. Next slide. Another thing that on our advocacy side of the house that we've just done is we actually just filed a lawsuit to protect wolves on Prince of Wales Island. And this is actually a really good example of the type of work that we do. So as a little bit of background, the wolves on Prince of Wales Island are genetically distinct. And in 2018, it was estimated that there was only about 170 wolves left. In 2019, ADF&G changed their management strategy and opened an unlimited 2-month season for trapping wolves and essentially asked the trappers to moderate their efforts. During that 2-month window, 165 wolves were killed. That's very concerning from a biological standpoint, from ecosystem standpoint, and also just the survivability of these wolves. So this past summer, we twice petitioned Alaska Department of Fish and Game's commissioner to close the upcoming season to prevent overharvest and significant damage to this population of wolves and to give them time for the numbers to rebound. The commissioner denied both of our petitions. Next slide. Just a couple of weeks ago, there we go. A couple of weeks ago, at the end of October, ADF&G testified before the Southeast Alaska Subsistence Regional Advisory Council that they were planning to open a wolf hunting and trapping season this year. But when asked, they actually refused to share the most recent wolf population estimates. So we were very concerned that so many wolves are trapped last year that there's not enough if the trapping management strategy doesn't change. So we have actually filed a lawsuit now, since our petitions were denied, requesting the closure of the 2020-2021 state wolf and hunting and trapping seasons on Prince of Wales Island. This will allow time for that population to rebound enough that it could allow for a sustained harvest within the Board of Games population objectives. 150 to 200 wolves is what they're shooting for. So this is just an example of an advocacy issue that we just did. We think it's very important to protect Alaska's wildlife and ecosystem-based management. We're not against hunting and trapping per se, but they need to be done in sustainable and ethical manners. So, next slide. There we go. Okay, so part of our education program, ever since the pandemic started, we have switched our Wildlife Wednesday series to an online platform, and which is great because we've been able to record them, just like we're recording this one. And so if you've missed any of our prior virtual Wildlife Wednesdays, they are available on our website. They cover topics from belugas and bears and humpback whales and seabirds to how to be an ethical wildlife photographer and what it's like to be a wildlife veterinarian. And there's also a couple on there kind of focusing about what the Kenai rule is. So you can also go and look at those and learn a little bit more about that. And then we're excited, some of our upcoming presentations. In 2 weeks, we have another one. It's about harbor porpoise. In December, we have one that's with the Alaska Sea Life Center, kind of a behind the scenes. And then in February, the author of the book In Wild Trust will be speaking with us. So enough about Alaska Wildlife Alliance. I want to go ahead and introduce everybody to Valentina Melica, and she is going to be talking to us tonight about hormones and whales. Take it over, Valentina.
Okay, thank you so much, Mandy. It looks like there might be a little delay from when I switched the slide to when it showed up. So I'll try to keep that in mind during my talk. But thank you all for coming. It's good to see you all. My name is Valentina Melica, and I am a PhD candidate in fisheries at University of Alaska Fairbanks. Before we dive into my research, I'm going to introduce myself a little better so it doesn't feel like you're like just a stranger is talking to you. All right. So I was born and raised in Italy. I'm originally from a town in the northeast part of Italy called Treviso. It's located by Venice. I got my bachelor and my master's degree from the University of Trieste, as well, again, in Italy. And I focused my master thesis on reproductive ecology of polyps of moon jellyfish. During my master, I had the travel bug, so I started spending semesters abroad, and specifically I spent one semester at Appalachian State University Boone, North Carolina. Oh, there's a laser pointer somewhere too, right? Maybe. And then another semester, sorry about that, and another semester at Western Washington University in Bellingham, Washington. And then this is where I started like being interested in marine mammals, and I started getting some research experience working on behavior of arbor seals in response to anthropogenic activities. This had brought me to the desire of getting my PhD. And I started this educational path 2015 with a scholarship at University of Alaska Fairbanks Ocean Science in the Juneau Center. Then I've been living in Juneau and embracing the great outdoors of Alaska as well as the Alaskan life. Okay, so this was a little bit about me, and now I'm ready to share with you some of the results from my PhD work that focus on hormones in whales. And this is an interesting comparison as I use very, very tiny molecules to gather some very interesting insights on some of the biggest animals that have ever lived in our oceans. Whales are a charismatic megafauna. They play a very important role in the ecosystem. They are the ultimate recipient of bottom-up driven system. They feed on lower trophic levels, which means they're highly susceptible in changes into the environment. Therefore, they can be considered ecosystem sentinels, and they can provide a lot of information or indication if something is changing in their ecosystem. Moreover, they're also a source of economic profit, and they are a tourist attraction. I live in Juneau, and here the whale watching industry has been booming, and therefore whales are very important for many coastal area along everywhere, but specifically here along the Pacific coast. There are also some species are also a source of subsistence food, and they're culturally important to Native people along the Pacific Rim. For my PhD, I decided to focus on two species that share an habitat in the East Pacific, the gray whales and the blue whales. So do you guys see my point? Maybe not.
Yes, Val, we can see it.
Okay, great. So here we have a gray whale, and here we have a blue whale. These two species of baleen whales, they have some unique features. As I said, they both live in the North Pacific Ocean, and both of them, for both populations, were driven very close to extinction during commercial whaling. Gray whales are known to perform the longest migration in mammals with a migratory route of about 14,000 miles or 22,000 kilometers. They're only found in the Pacific Ocean, and they're bottom feeders, or as I like to say, they're suckers. They suction feed from the bottom, and pretty much they eat everything they can keep besides mud and water that gets drained. On the other hand, we have blue whales, which are the biggest animal that has ever existed. They're cosmopolitan species found in every ocean basin. And differently from gray whales, they're very picky eaters, and they only feed on krill. In the Eastern North Pacific, this population share migratory corridors. Gray whales, which are the migratory corridors are indicated by lines. They are coastal animals that spend their winter months in the well-recognized breeding grounds along the La Baja California Peninsula, for them migrating during the summer months in their feeding area along the US and Canada West Coast, and in some cases all the way to the Chukchi and Bering Sea. Migratory grounds for blue whale are a little less well known, and here are indicated by the red arrows. Know that their habitat expands as far south as the Costa Rica Dome, and they have been sighted in the Gulf of Alaska, which is considered kind of like their most northern boundaries. We know that the Gulf of California is a recognized reproductive ground for this population, with many mother calf pairs sighted during winter months, as well as Southern California is a well-known feeding area where large aggregation of blue whales are sighted during the summer months. Within their habitat, these species are exposed to multiple sources of disturbances, both anthropogenic and oceanographic. For example, in Southern California, the migratory corridors for blue whales overlap with shipping along the Channel Islands, just outside of Santa Barbara. And this is like here you can see in this graph, you can see the shipping lanes, and they fully overlap with the migratory corridors of gray whales as coastal are very susceptible to entanglement in fishing gear and crab. And both these species, as I said, they feed on lower trophic level, which means if there's changes in their oceanographic conditions, there's going to be changes in the prey availability and prey abundance. And here in this map, you can see, for example, an example of change in the oceanographic condition. It's the sea surface temperature, also known as heat wave or blob, in the Pacific Ocean. One other thing that put these two species in common is there's a lot of that is not quite understand. A lot of research, especially post-whaling, has focused on photo identification, population abundance, behavior, and migration as well. But not very much on physiological process. All we know about physiological processes on these two species come from study conducted during commercial whaling. And why this is still valid information, there is the need for some updates on reproductive parameters, as well as in general knowledge on physiological processes in whale. And this is where my research fits in. The overall goal of my project is to contribute to updating this knowledge, and specifically focusing on two main reproductive processes to provide new data for, and those are reproductions and stress response. Before we dive into the depth of my research, let me give you a little brief overlook of how these two physiological processes work in the mammalian body. So we start with reproduction, and it all starts with some outside stimuli that can be changing photoperiod, pheromones, co-presence of males and females. This trigger a cascading of reaction in our body that involves multiple organs, from the brain to the final target organs, which are the gonads. Here, this leads eventually to gametes production, and in case of conception, to pregnancy. Hormones act as messengers between each of these parties in this cascades of reaction. And specifically, what we're interested in what I was interested in for my project were the two hormones produced by the gonads. So I was particularly interested in investigating progesterone, which is secreted by the ovaries and is involved in ovulation and in sustainment of pregnancy, as well as testosterone, which is secreted by the testes in males and is responsible for spermatogenesis, as well as development of secondary sexual characteristics. Now let's take a look at the stress. It works in a similar way. We start with some outside stimuli. We have noise, we have a change in the environment, and so on. That triggers a reaction cascade that leads to a response. Now, many of you are familiar with this type of response, their fight or flight, that it's mainly triggered by an acute stress, for example, the presence of a predator. Physiologically, this is usually detected by an adrenaline rush. But sometimes when we have a stressor that is prolonged or is repeated, we are in the circumstances of chronic stress. the chronic stress is dealt a little differently from the acute stress that is dealt with a fight or flight response. Chronic stress is dealt mainly by the adrenal glands that are located by the kidneys. The adrenal glands release compounds whose main job is to keep the body going in a prolonged stress situation. And to do that, this trigger processes that will access energy storage, for example, adipose tissue or muscle, to meet that energy demand. In severe cases, or in general over a prolonged time, this type of stress can have some serious detrimental effects. It can cause poor body condition. It can debilitate the immune system. And in severe cases, it can cause reproductive failure. For endangered population, this could overall lead to a population decline. As I said, hormones in these processes act as messengers. Therefore, there can be some sort of biomarkers. A biomarker is a measurable indicator of some biological state on condition. And we use biomarker to detect when there is a change, when there is a change from a normal state to an altered situation. You might wonder, where do you find hormones in whales? Well, they're everywhere, but in my case, I decided specifically to focus on hormones, cortisol, corticosterone, progesterone, testosterone, that belongs to the class of steroids. Steroid hormones are lipophilic, which means they love fat. And whales do have a lot of fat, luckily for me. Whales have a very thick layer of adipose tissue known as blubber, which represents an important energy storage. It's involved in thermoregulation as well as in buoyancy. But it's right there in this adipose tissue that hormones, that steroid hormones, can accumulate once they are secreted during physiological phenomena. So we can extract and we can measure the hormone concentration in whale blubber. And this is how we do it. This is how I did it for 5 years. Basically, here we have a blubber samples from, this is from a stranded gray whale. And as you can see, we have the skin over here, the blubber tissue, and then over here, that's where the muscle will attach. Hormones are extracted by hand squishing. I have no other terms that will better describe this process, using multiple organic chemicals in the process. And then from this extract, you can measure the concentration of hormones using a technique called enzyme-linked immune absorbance assay. We're just going to call it EIA. The EIA is a technique based on antigen-antibody competition. We have an antibody that is able to recognize the hormone and measure the concentration against a standard curve. This technique is pretty widespread. I like to point out that some of the antibody tests of coronavirus are actually EIA. And also, thanks to this technique, Rosalind Yalow won the Nobel Prize in 1977, and she was, I think, the second woman to ever won the Nobel Prize. One other really benefit of this technique is that they can be used on a wide range of wildlife species, making it pretty easy to use, and it's relatively inexpensive. Coupled with this technique, I also had access to a lot of information about the animals. So for blue whales, I was lucky to collaborate with Cascadia Research Collective and the CCMAR Centrum in La Paz, Baja California, that has provided blubber biopsy from blue whales collected in two main important grounds: the reproductive grounds in the Gulf of California over here by the purple, and the feeding grounds along the US West Coast. even more, they provided, they let me access their photo ID catalog so I could gather some information about the animals. And I was particularly interested in categorizing the animals based on their age class, identifying adults as animals that had a sighting history of at least 8 years. 8 years is the average recognized age of sexual maturity for blue and for gray whales. So I use that cutoff, that threshold, to identify adult animals. Whereas I called calf or immature all the animals that were of known age, so we knew exactly the year they were born and we knew exactly what was their age at the time of sampling, and that age being less than 8 years old. And then for female, I was also interested in learning about the reproductive states. Specifically, I was interested in knowing if there were any pregnant females, females that were sighted with a calf the year after sampling, or lactating females, females that were sighted with a calf on the year of sampling. I did the same thing also for gray whale. For gray whales, I had the honor to collaborate again with Cascadia Research Collective, NOAA, Southwest Fisheries Science Center, and the Macaw Tribe Marine Mammal Program of the Olympic Peninsula that has provided a total of 106 biopsies that were collected along the Pacific coast while the animals were in their feeding grounds. And similarly for blue whales, I access photo ID catalog to categorize the animals in their age class, reproductive states. So the two main objectives of my work were to develop biomarkers for reproduction using progesterone and testosterone, and then develop biomarkers that will allow us to detect ongoing stress response using testosterone in gray whales. Let's start with the first focus on reproduction first. For this part, I had two main questions, and we're going to start for the first one. We know that in pregnant females, progesterone tends to be elevated because progesterone is the main hormone secreted during pregnancy, and it is responsible to sustaining this physiological status. So I wanted to know if I could actually use progesterone concentration to determine the status of a female whales. To do that, I measured progesterone concentration in blubber, and then I developed a model that will allow me to calculate the probability of a whale to be pregnant based on her progesterone concentration. So in this graph, we have on the x-axis, we have progesterone concentration over here. And on the y-axis, we have the probability going from 0 to 100 of a whale being from going from pregnant to pregnant. I highlighted here the 95% line, and all these dots are single whales. So as you can see, all whales over here above the 95% line, they have higher than 5% probability of being pregnant. In number, this looks like that. I was able to identify a total of 15 gray whales pregnant and 10 blue whales. Now, out of this, for the gray whales, only 4 were confirmed as pregnant. And the one that were not confirmed, that I called presumed, only 4 were resighted the year after sampling, and they were not accompanied by a calf. This provides us some information about the potential of about the rates of calf loss or the potential of reproductive failure. Similarly, for blue whales, the sample size was a little smaller, but I had a total of 10 pregnant whales. 4 of them were confirmed, and out of the 6 that were presumed, only 2 were resighted the year after sampling without a calf. This might be, in this case, we don't know very much about the breeding habitat of blue whales. So this might be an indication that there are alternative reproductive grounds that we don't know about, or they're not as well studied as the Gulf of California. Now let's take a look at the males now. So testosterone is known to have a cyclic pattern in adult males of any of many wildlife population. What that means is that it tends to increase as the animal approaches the breeding season because it's getting physiologically ready to mate. But this has never been really tested in many whale species. So what I wanted to know is whether I can use testosterone to help estimate the timing of reproduction, looking for seasonal changes. And for both species, so both this bar graph shows season on the x-axis, and then on the y-axis, it's concentration of testosterone. And for both species, you can see testosterone is higher in animals that are sampled later in the year, in fall for blue whales, fall for gray whales. And this is an indication that this is when these animals are getting physiologically ready to mate. At the time of year, both the species are still in their feeding ground. And this suggests that for both blue whales and gray whales, mating really occur at the beginning of their southbound migration as they start heading south towards the tropical water. So from this first part of my research, I learned two main take-home message. We can use progesterone as some sort of pregnancy test for whale, and it can help us detect the number of pregnant whales in a group, detect pregnancy, and this can lead to produce and to calculate much more detailed and accurate pregnancy rates. Testosterone can be very informative about the breeding times and breeding areas of whales population. And this is particularly crucial for a population we really don't know much about, such as blue whales. Okay, now before we switch gears and we talk about stress, let me just remark one more time why we are so interested in physiological processes in whales, and particularly stress. Whales fits very nicely in the One Health concept, which is a concept that highlights the interconnection among human, wildlife, and environmental health. What this means is that everything is connected, and healthy whales means healthy oceans, healthy fish, and therefore healthy food. And whales in this case can act as a good model organism. If there's something wrong, if we can detect an alteration in the health of the whale population, this is probably indicative of something happening at the environmental level, and that will eventually affect us as well. A little bit of attention is being paid to the fact that there's been a lot of an increased mortalities of whales, especially over the past 4 or 5 years. We have heard a lot about mortalities, more stranded whales, mass stranding. And this unusual mortality events, or UME, they make us wonder whether there are underlying causes that are connected to it, and if there's other causes that we should worry about at the ecosystem levels. This is a particularly this is very applicable to gray whales. Gray whales have experienced a UME, an unusual mortality event, in the 1999-2000. In those years, in that period, more than 600 whales were found stranded, and that has caused a significant decline in the population. More recently, 2019, a population is a rate of mortality. And therefore, actually, NOAA had issued a second UME for this species for the 2019-2020. And in this graph, you can see, in the bar graph, you can see the number of stranded whales found over season, over months. And in the map, you can also see where those trending were found. And if you remember from the initial slide, basically mortality is affecting the entire migratory area for gray whales. While no common cause was found to explain this high mortality, but in 1999, and it seems like also for some individuals in 2019, many of these individuals were found in poor body condition. Poor body condition can be connected again to that stress response to chronic stress that I told you about before, where we have compounds whose job is to keep the body going, and therefore they will keep accessing energy storage that will eventually lead to emaciation and poor body condition. So from this is where I formulated my question, whether we can use these hormones to detect an ongoing stress response. And to do that, I needed to build a comparison between healthy and stressed animals. Healthy whales, I consider healthy whales live animals, which were sampled through a biopsy, which is an example here is the picture of the blue whale with this biopsy arrow, right, hitting it right there. Didn't hurt her, don't worry. It's just like, I don't know, like a mosquito bite. Compared to stressed whales in this case, compared to stranded animals. Before, excuse me, before I can fully build this comparison, I needed to check something else. I know that so far I made stress appear a very bad thing, but in reality, stress is not only bad. We do need a stress response. We do need those compounds. We do need adrenaline. We do need cortisol and corticosterone, which is another stress-related hormones. They increase the awareness. They're related to growth and reproductions. They have their role in the natural state of our body. So I needed to understand first, how do they naturally behave in healthy animals in their different, let's say, everyday life. Generally, I noted that females tended to have higher concentration of hormones. And so I questioned whether maybe reproductive status was a particularly significant factor in those hormones concentration. And soon enough, this is what I found. First of all, I talked about cortisol and corticosterone. These are two hormones that are very similar, and they do the same thing, let's put it that way. But one of the main interesting facts is that to each species its own. Cortisol is considered the stress response hormones for mammals, and corticosterone is considered hormones for birds, for example. But this is interesting that I found that cortisol is a better biomarker for gray whales, whereas corticosterone is actually more appropriate to identify an ongoing stress response in blue whales. Here, these graphs show the concentration of these hormones for each of the species in response to a reproductive status. And not surprisingly, lactating females in gray whales, tends to have higher cortisol concentration. Whereas for blue whales, both lactating and pregnant females seems to be a little more stressed than everybody else, which is, again, not surprising. There's quite a lot going on in their life, and particularly for whales, lactation is a pretty costly process in terms of energy. So now, knowing how the hormones generally behave, I was able to go back to my original question and build that comparison between healthy and stressed whales. And this is what I found. Generally, again, I found confirmation that cortisol is a better marker, is a better biomarker for gray whale, whereas corticosterone be more indicative of stress response in blue whales. For both the species and for both these hormones, stressed or stranded animals had much more elevated concentration of these hormones. And you can see that in this bar graph. Here are the stressed for both like gray whales and blue whales. For gray whales, I was able to take even a deeper look. For gray whales, and here I have to shout out a big thank you to the Stranding Network, and particularly the Alaska Stranding Network, who has been key in providing a lot of samples from stranded gray whales. I had a bigger sample size. I had a total of 47 individual, and although these different individual perished to different cause of death. There were some individuals that perished due to entanglement, some to trauma, which could be ship strike, some were emaciated, so I consider them nutritionally stressed, some were part of the UME, and some were killed during a hunt by whales, for example. So I was curious to see if the cause of death could affect hormone concentration. And interesting enough, I found that animals that perished to trauma or were particularly emaciated had much higher concentration of cortisol. What this means is that likely these animals didn't die straight away. Rather, they lingered, or in the case of the nutritional stress, they were probably not able to feed for a very long time, proving that, not proving, but supporting that cortisol can be an indicator of chronic stress. So what this all means and why do we care? From study like mine, we can gather a lot of innovative insights on reproductive timing and grounds for a population we really don't know much about. Again, we can use progesterone as a pregnancy test and calculate pregnancy rate as well as parturition rate and investigate further phenomenon like pregnancy failure, for example. We can also understand a little more about their timing in mating and maybe think about conservation and protection efforts that will ensure limited disturbance in that time and in those areas. Also, using stress hormones, we can build long-term efforts to monitor the effects of change as well as the effects of human activities on survival and reproduction of this population. And all this information can be used, and it's vital to develop a much more accurate population models that will predict population growth and trends. Okay. I would like to thank my funding and collaborators that have made this research possible. The Fulbright that has allowed me basically to start my PhD, University of Alaska Fairbanks, University of Alaska Southeast, Alaska IMBRE, Office of Naval Research that have funded this project. And then all my collaborators from Cascadia, CCMAR, from Cascadia Research Collective, CCMAR, NOAA, the Marine Mammal Tribe, I'm sorry, Tamaka Tribe. It is a 7:44 p.m. Sorry about that. The tribe that have provided, and the Marine Mammal Stranding Network that have provided all the samples that I use in my research. And with that, I will be happy to take any questions if you guys have any. And thank you for listening.
Thank you very much, Valentina. That was great. Really appreciate it. And folks, I don't know if you have on my toolbar, I have a reactions where we can give her like a little applause. So we can all say thank you. now's the time to go ahead, and if you have any questions for Valentina, let's go ahead and put them in the chat function. You can send them to me as the host. In the meantime, I want to say, thank you to Valentina. She volunteered her time to put together this presentation, and it does take a lot of time, and so we're very appreciative of that, and that you're willing to share your knowledge and your research with us. We want to thank all of our members of Alaska Wildlife Alliance. Your donations allow us to put on these types of educational seminars. And then just everybody who is on tonight's presentation, or if you're watching this later as a recording, you clearly care about wildlife, and we are so thrilled to have you with us. So while folks are getting a moment to put in their questions in the chat, Valentina, I've got a question for you. I found your comparison of when you had the different reproductive states and the stress levels. I expected that lactation and pregnancy would be very stressful, but it was very interesting to see the difference of how it was fairly low for gray whales, but really high for blue whales. Do you have any theories why pregnancy appears to be so much more stressful for blue whales?
Yes, I do have some ideas. So it was very high for blue whales, and most of the pregnant blue whales that I had in my sample size were the late phase of the pregnancy. So it's possible that that might have been a longer time for the whereas I suspect, and I'm not able to confirm, that some of the pregnant gray whales, might have been in our earlier stages of their pregnancy. One other thing to consider also, and these are my hypotheses. All of the gray whale samples that I had came from the feeding ground. And so they were a mix of animals that were on the way up north and animals that were on the way down south. What this means is that some of the animals have been feeding for a long time and some have not. And so it is possible that the overall blubber layer or the overall adipose tissue was of different thickness, and blubber is a very complicated tissue, but I want to say thickness, and maybe the lipid content might have been different between those stages. And it's something that I would love to look at.
Oh, great. Yeah, that actually, your just final comment there. Someone asked a question, what is your next step in this type of research?
Oh, for this type of research, I hope, I think the next step for this would be to incorporate additional indicators. Hormones are one example of good bioindicators, but there's many more. One, I mentioned it, and that is lipid content. It will actually be very interesting to look at the lipid content to understand a little more about the nutritional status of kind of understand that will help us better understand how the hormones do concentrate over tissue. I would also be very interested in considering increasing the sample size. I think for stress related studies, it is ideal if we can actually have a point source of stress and compare animals that are exposed to it and they're not exposed to it. However, that is not very feasible for whale population, as they might be exposed to it at different time. But knowing that lactation and pregnancy are very stressful phenomena, or are very stressful time of life of a whale, we could compare that as the most stressful situation for a whale to other statuses, and that will give us a better understanding of how they cope with stress. And then a timeline, look at, and that, again, with an increased sample size, consider oceanographic factors such as sea surface temperature, PDO regime shifts, Pacific decadal oscillation regime shift, El Nino, La Nina pattern, to see if there's any correlation or if there's any relationship at all with change in stress hormones.
And that's actually a pretty good segue into another question. We're talking about environmental factors. So the question is, the UME animals had lower cortisol than nutritionally stressed. Does that indicate to you that the diet is likely not the cause of the UME?
It's possible. You guys can see, okay, yes, can see the whole, not all the UME animals where in poor body condition were found, which it might be an indicator that there might be other underlying cause rather than necessarily not access to prey or lack of prey abundance. Keep in mind, whales have been increasing exponentially. Actually, they have been growing as population. Therefore, dependent behavior and density dependent effect that is happening, and maybe we are missing that, and that could be even connected to the UME.
So you froze up just for a second. Did I hear you correctly that
sorry.
you said it might be a density dependent effect?
Yes.
And we're just missing that? Okay.
Yes, correct.
Just want to make sure I understood. And while we're on this slide, there was a question where someone was surprised that entanglement wasn't associated with a higher level of stress. Does that imply a relatively quick death rather than prolonged suffering?
Yes, that is what I think that implies. That is my following hypothesis for that. However, I do understand the confusion, and I was surprised at the beginning because in other species, similar studies have actually shown that whales that have been entangled tend to accumulate more cortisol. And that was a study done on North Atlantic right whales. But I do think that for the specimen that I had for that specific category, one was a live stranding. So that animal got entangled and stranded straight away, or like died, I think, within hours from the stranding. So it's possible from the entanglement. So it's possible that really the process wasn't long enough to actually have those hormones to accumulate. One other thing that it might help explain this. One of the animals in this group was a juvenile, and I'm not quite sure, but I would hypothesize that probably the blubber layer not as thick as it would be in an adult individual, and that could potentially affect also the concentration of hormones.
I have to wonder. Yeah, I wonder if another potential explanation is, this category, this slide is talking about the cause of death. And in my experience working with marine mammal veterinarians, an animal may be entangled for quite some time, but it's not actually the entanglement itself. There's other factors, for instance, poor body condition that they've carried because they've carried gear for so long that it's reduced other things. And so then it's kind of the other thing is what the veterinarians, during a necropsy might say was the cause of death, but that thing might have been caused by the entanglement. So this one might be a little challenging to tease out based upon the way the veterinarians are designating cause of death. So, but really good question there. And we have another one. Are you still able to sample this year when ocean noise and traffic are lower because of COVID shutdowns?
They did. I didn't. This was my final year. I actually defended my dissertation about a month ago. So I'm on my way out, but I do think that all my collaborators have been doing field effort exactly for that same reason, because this is a new unique opportunity to actually gather samples in a year where the disturbance might be much less than normal.
Yeah, it would be really fascinating to kind of see, last year's sampling, this year's sampling, and next year's sampling, and to see if there's a dip in those stress levels of responses. So any more questions? I'm not seeing anything come through the chat, so I want to make sure, folks, this is now your chance to get any last lingering questions in there for Valentina before we let her go. Here's a comment that, glad to hear that some sampling did get done this year, yay. So that's good to hear. And then some congratulations on your defense.
Thanks.
And then a comment that this was a great study, thank you.
Thank you all.
So of course, everybody can use this as a way to share your kudos with Valentina too. It doesn't just have to be your questions. And then, if no more questions come in, one of the things I always ask at the end of our Wildlife Wednesday seminars is we'd love to know where folks are joining us from. We're quite surprised that we sometimes have international folks from all over the US. So we're reaching more folks, and more people care about Alaska's wildlife than just Alaskans. So, I'm not seeing any more questions come in. So again, a round of applause. Thank you very much, Valentina. We're so honored that you shared your research with us. And I'm going to go ahead and stop recording. And again, if you will just let me know where you're joining us from before you sign off, that would be great. Thank you.
Thank you all. I appreciate it. Thank you for listening. It was great to be able to share my research with you all.
And you're getting a bunch of kudos and thank yous coming in.
Oh, that's great. Thank you. Oh, and if anybody has any question, here's my, where is it? Here's my email in case they want to reach out to me later as well.
All right, there we go.